Assalamu alaikum. In this section, we discuss about column chromatography. In the previous section, we discussed liquid chromatography. Okay, now column chromatography. A column chromatography in which we use stationary phase as a solid as well as liquid. If we take a stationary phase solid, it's a dry fine powders, and if we take a liquid, it's a wet granules. Now we discuss about what is column chromatography and how column chromatography is prepared. First, we discuss about the chromatographic column. Now, imagine that it's a syringe-like structure. It's a syringe-like structure. Now, we call it a column. And how we prepare it? First, we wash the column with polar solvent, non-polar solvent, then water, then dry. First we wash a column, then we place a cotton on the valve side. We place a cotton. What cotton do? That's purity and control flow. That maintain a purity of the component and control the flow of the component. Then we place a sand layer that maintain the equilibrium. Then we place a stationary phase. Then we again place a sand layer. Then we place a component of the mixture. Then we place a mobile phase. Now after that we prepared a column. Now we open a valve. That is a valve. We open a valve and that eluate is secreted from this column. Okay, now that is the preparation of the column. Now there are two types of column that is prepared by available as the prepared of the column as automatic prepared, and also a column that we prepared by ourselves. Now the automatic column that is commercially available is 50 ml bullet that can be used. These are in this paragraph it's right about how to prepare it. After that we prepare the column. The mixture is separated. How mixture is separated? Mixture is separated on the base of their polarity. Now the different bands appear on the stationary phase on the basics of their nature. Because of the stationary phase is polar, a polar component can be absorbed on this stationary phase. A band appear on the stationary phase. After that we can determine the its component nature and its quantity also. Okay, now what is called a development. Now chromatography and development is similar. There is another name is development. In liquid chromatography, the process of separation of a mixture into its component will take place as the mobile phase flow through the steady phase is called development. It's same definition like chromatography. Okay, now there's other phenomena we call its elution. What is elution? Now the carrying of the component of the mixture by the mobile phase through the process is called elution. Now if we discuss in detail, if we prepare the column and our component is flow through the stationary phase and the solvent, the mixture that is separated in the last, this all process this sweep through the component and stationary phase we call elution. And eluent, the solvent we use, the mobile phase we call eluent, and the mixture in the end that is secreted or separated we call eluate. How it's separated, how its bonds appear in the column chromatography or stationary phase. Now on the basic of their polarity. If a polar compound is absorbed, a band is appeared. In the less polar compound is absorbed, it's appear on different sides, and more polar on different sides because of their attraction between polar and polar compound. Okay, next we discuss about chromatogram. A chromatogram is that um, a, a result that is appear on the graph or something like that. We now about which kind of nature of this compound we call this chromatogram. Okay, now we use a detector, different detector, to determine the which nature of this compound is that. Okay, if we discuss a definition, a plot of the detector response as a function of eluate volume or eluate time is known as chromatogram. Because of this, this is a separation on the basis of their eluate mixture. Now we can determine this eluate. We detect this and determine which nature of compound is it. Okay, now there's another important phenomena, that is gradient elution. Okay, before going to this gradient elution, in column we can't use a polar solvent because of its absorption to the stationary phase or miscible in the stationary phase. If we use a polar solvent that is absorbed in the stationary phase or miscible in the stationary phase, our component is can't be separated. No space is available for the separation of our component. That's why we use gradient elution. And what is gradient elution? Gradient elution is a process in which the composition of mobile phase is gradually changed due to the development of chromatic dye. Now mobile phase is gradually changed. Now we in the previous section we said that we use a mobile phase as a binary solvent. Now in this technique we also use a binary solvent, but the concentration of the polar and non-polar we use different and we change it different. Now first we use more less polar compound and less polar compound. For example, we use a 95 and 5 ratio. We use more 95% less polar and 5% polar. The polar component that is separated with short retention time. What is retention time? The time required to sweep a component from the mobile phase or the stationary phase. And after that, after the eluate is obtained, we use we increase the concentration of the polar and decrease the concentration of less polar. Then the polar compound is separated. Now the solvent we use a gradient elution must be completely miscible with each other. What does this mean? It means that we use two solvent in a mobile phase. We call it binary solvent. These two solvents are miscible with each other. Okay, detector. Now we can detect a fixed volume portion. Now we can detect eluate by fixed volume portion because it's a liquid. We use a different fixed volume ratio or a fixed time ratio. We also determine it. How we can determine? We take a spot on TLC and check the components after 20 to 30 minutes on TLC. After that we check that spots is appear, separation is now the component of this spot is appear on the TLC. We use different detector methods to detect the component of the mixture. Now in this we discuss detector, different types of detector we can use. Some detectors are automatically connected to the column chromatography. For example, we use UV or visible absorbing cells. In UV visible absorbing cells that is connected to the column and that is continuously monitor the basis of the separation of the component. But if our component is non-absorbing compound, then we first pass into our reactive column that convert the non-absorbing compound into absorbing compound, and then we detect it and monitor it which kind of compound is that. There's also detector which is continuously monitored, and some we monitor as individually by assessing a TLC that we discuss now. Okay, we can also connect a mass spectro technique with column chromatography that determine the separation information about the separation of the compound or the molecular weight of the compound.